Welcome to our talk today. Before I focus on today's presentation and the speaker, I should say it's a very happy coincidence that our first colloquium talk of our thematic program, which is abbreviated as TDA Party. The full name is a little bit long, so we use this funny sounding abbreviation, TDA Party. It's a nice coincidence that actually the colloquium happens exactly in the middle of the thematic program. It's the 4th week of a 7-week program, and this is the midday of this week. So with that being said, there has been some other components that you can look at if you're interested in the broader scope of the program, and there'll be some more. Now I shift my attention to today's colloquium talk, and it's my pleasure to welcome and introduce Professor Ezra Miller from Duke University. His list of accomplishments is too long, and instead of me telling you why he's a successful mathematician researcher, I give him more time to demonstrate it live. Instead of listing those accomplishments, I just want to point out a couple of matters that outline the broad scope of his research. So he did his bachelor's at Brown University, and reading from the brief bio available on the website, the bachelor was a Bachelor of Science in Mathematics and Bachelor of Arts in Music with a focus on theory of composition in 1995. Then he followed his academic trajectory with his PhD program from University of Berkeley, University of California at Berkeley, 2000, followed by a couple of postdoctoral researcher stays, one of which was at MIT, then one at Berkeley again, if I'm not mistaken, MSRI, and then he started his tenure track position at the University of Minnesota. And from 2009, he went to Duke University, and he has been a prominent researcher in various domains of research. We just received a direct message from somewhere. Some of those research areas include combinatorics, commutative algebras, computational geometry, combinatorial game theory to high-dimensional statistics and computational neuroscience, and some more. And today's talk particularly focuses on asymptotics for biological data in non-smooth spaces. And I should say, on the two personal notes I'm allowed to leave. One of which is like the first time that I really found him a good speaker to enjoy was not the first talk of him that I attended, which goes back to 2014. It was very nice. I don't know who does that, but I give the credit to you.
Made that noise before.
The first time that I really found you as an engaging speaker was a couple of talks that you gave in Montreal during your research stay.
Oh, yeah.
And they were very pedagogical and allowed me to actually get a first-hand exposition to connection to TDA. That was the first personal note. And the second personal note is that the speaker is just fresh out of water. I can elaborate on that later. Please go ahead and start it.
Thank you, Kava, for that generous introduction. Just a question: how many people are online? Are there people online?
There are. Currently, we have 8 participants. Okay. 2 of which
very good.
Must be ours.
Okay, so if people online have questions at any time, or if something that's online, if something I'm doing in here is not clear online, it's important to let me know. So for instance, I assume everyone can hear me online. If not, then please do tell me so. I turned on the microphone and everything. Okay. So I don't know the backgrounds of everybody attending this talk. I know that there are mathematicians in the audience. I don't know whether there are biologists in the audience. There are biologists in the audience. Very good. So I intended to say something. So it does get pretty mathematical at some point, but it starts off pretty biological, and then sort of the math can be soaked up at whatever level your mathematics happens to be. So let's just, so this is the main results I'm talking about are joint work with Jonathan Mattingly, who's at Duke, and Tran, who was my graduate student at Duke and then went on to a postdoc at Göttingen, but then left academic math and is now now works at Deutsche Bank. But here's a summary of what's going to happen here. So these things are listed across the top, so you can see them go across the top like that. I don't know what that sound was. This first slide takes quite a while because it has a lot of pictures and things, but all the rest of them are just 1 or 2 slides and so on. So if you don't get worried that we've only gone through 1 slide after the first 15 minutes. So I'll start by talking about some data, and then I'll talk about what the central limit theorem usually says, and then go into some details about what you do when you try to do central limit theorems when you don't have a linear structure like a vector space sitting around. What do you do instead? And that's what this talk is basically about. First, there's the smooth setting, and then there's the singular setting, and then when you do the singular setting,
There will be somebody online.
oh, it could be somebody online. That's true.
Don't see
I'll just continue while people figure that out. But you need various hypotheses, and this is where the mathematics comes in. Is what spaces do you need, and how do you do things, and so on. Okay. Yep. Okay. Nonlinear data. So the idea is biological data are often sampled from nonlinear spaces, like the data. What happened there? Oh,
I think it's a restart. Like the online people still have your screen, but I think it's coming back.
So the physical projector has done something.
Yeah.
Okay, so I'll just talk for a little bit. Well, I'll talk while it reloads. So biological, oh, there it is, it's back. So biological data are often complicated. Those who work in biology know this already. So here are just examples of data that are not just points in vector spaces. So angles, points on a circle, right? If you're looking at knee motion or elbow motion, that gives you a point on a circle, right? And if you have a wind direction, that's not biological exactly, although it comes up from biology, but that's also the direction is a point on a circle. If you have directions in space, then these give you points on a sphere. So wrist or ankle motion, right? So if you look at wrist motion, you're basically picking a point on a sphere around the center of your wrist. Ankle motion, same thing. But it comes up in other kinds of medical imaging and various other places like this. So here's an example of a hippocampus surface. And one way to represent this surface is by sort of drawing a medial axis, which would be sort of a center line, and then saying how you go out from that center line to reach the surface. And so then describing how you go out from that center line at each of these points, there's sort of 6 points along the interior here, and then maybe 18 points around the edge. And each of those has some data attached to it. The ones around the edge have 2, maybe 3. That's definitely not here. 2, maybe 3 sort of vectors pointing out into space, telling you where the surface is, and there's some radius that comes with it. All told, each data point here, so here's a data point. This is just one hippocampal surface, the skeleton representation. There's another skeletal representation of another surface, and this is when you superimpose them, you can see the little differences between them. That's the point. And each data point is a point in a 210 dimensional vector space, but they all lie on this 162 dimensional sub-object. And this 162 dimensional sub-object is a manifold in this case. Here's the manifold described to you quite specifically. More recently, they have made this even more complicated by sort of allowing the representation to sort of grow out from the center. So I just wanted to mention that because I gave this talk and I gave this slide, I'm sorry, in front of Steve Pizer last year, and he said, we've done more recent stuff. So there's more recent stuff. Shapes. So what is a shape? So a shape is think of it as a landmark shape where you have now a finite set of points. You've got maybe some mandible or some cell or something, and you've marked a bunch of points in there, and you have a different cell, and you've marked a corresponding set of points. And so what you have is each shape is summarized by a finite set of points, but you want to say that this set of points and that set of points are the same. So they're the same up to rotation, and maybe they're the same up to scaling or some projective transformation, depending on how you took your images and so on. So the point is, what you're really doing is you're looking at, if you have, say, in R3, if you've got a bunch of points, then what you have is a D by N matrix. It's a 3 by N matrix. So you have N points in RD. And then you mod out by some group of transformations. And this gives you a quotient of a vector space And that quotient is an algebraic variety, and that algebraic variety is often singular. And if you want to do inference by sort of looking, each data point is a point in that variety, and then you want to do statistics with that. Diffusion tensors, each diffusion tensor is a positive semi-definite matrix, and you could have huge families of these things. These are DTI images of a brain, and the point is that at each point in the brain, each point in 3 space, you can look at how water diffuses out of that point. And when you're going along a nerve, it tends to diffuse faster along the nerve than out of the nerve. And then these, so in each point you have a direction, or you really have an ellipsoid. And so at each point you have an ellipsoid, and so you have this whole family of these ellipsoids. That's what this is really a picture of. The color is telling you in which direction the ellipsoid is pointing. And this gives you some proxy for how things are flowing in the brain and connections in the brain. And this gives you data that you need to mine if you're going to figure out what's the correlation between essentially how someone thinks and what it looks like. Let's just leave it at that. Trees. Biology has all sorts of trees. There's phylogenetic trees. These are lung airways. No, these are lung arteries. These are arteries in the lungs. Now, in the lungs of what? These actually turn out to be arteries in the lungs of a fetal mouse. So the mouse itself was only about maybe less than a centimeter, and so the lungs are just maybe a millimeter or 2. And then what you do is you have a microphone and you fill those lungs with some fluid, and you image them, and you get these detailed pictures. This is oh, right, I have to do it online. This is a normal pair of lungs, and this is a pair of lungs where the mouse has congenital diaphragmatic hernia, which causes various problems. And the idea is to quantify the difference between these 2 sets of lungs. That's the idea. Here's another tree. There's actually 4 trees. It's not connected. But anyway, this is Steve Marin's brain. Steve Marin was a co-author of mine on a study that we did. And he was one of the participants. And so we like to put his brain up. This is actually the arteries in Steve Marin's brain. And you can see this is a complicated 3-dimensional thing. And the idea is that we've got 100 of these, and we want to do statistics with that. How do you do statistics with that? That's the question. These are phylogenetic trees, right? This is a phylogenetic tree for some crickets. Full disclosure, this is my wife. That's where I got the image from, O'Neill. So this is just a reconstructed tree, but how is this tree reconstructed, right? So there was some information that we had, genetic information or maybe shape information, right? This is morphological information. And you want to know, how do you reconstruct a tree with this sort of information? And then maybe you have a family of these trees and you want to do inference with that family of trees. Yeah. So here's an example of a space of trees, the phylogenetic tree space of Valera, Holmes, and Boekman. So suppose that you have so what is a phylogenetic tree mathematically? It is a rooted metric tree with n labeled leaves. Yeah, so rooted means that, so in these phylogenetic trees, that's the most recent common ancestor back there. That's the top of the tree, if you like. In this picture, it's the top. On the previous picture, it was on the left. And it's metric in the sense that you have some idea of how long the branches are. They can be measured in time. They can be measured in SNPs, single nucleotide polymorphisms. They can be measured in various ways, but there's a notion of length. Where do you get distributions of trees? So that means you have more than one tree. Well, if you try to reconstruct a tree, it's such a complicated problem. There's no good way to reconstruct the tree that comes from this data. What you do is you sort of walk randomly on tree space until you get a tree that is you consider to be a randomly sampled from the set of possible trees in a way that's sort of sampled from a distribution that's dependent on how the data look. So the idea is you reconstruct a tree probabilistically. What that gives you now is a distribution of trees, that is a set of trees, and that set of trees is the reconstruction of the tree that comes from this data set. So the reconstruction of a tree is really a reconstruction of a family of trees, and then you want to summarize that family of trees. That's the idea. These can come from medical imaging, as you saw. They can come from evolutionary biology, where you have gene trees versus from a species tree. So those in evolutionary biology will know this sort of thing, but if you know the evolutionary history of a family of species, you have a set of species and you know they're actually their phylogenetic tree. If you pick a particular gene and you ask what's the history of that gene, it needn't look like the history of the set of species. That's because some genes are preserved under speciation events. Yeah, so the times at which genes, so the evolutionary history of a particular gene need not look like the evolutionary history of a set of species. So suppose that you have a set of species and you look at this gene and that gene and that gene and that gene, and each of them gives you a tree. What can you try to say about the evolutionary history of the species themselves rather than just the genes? Yeah. Okay. Sample space. So what is the space of phylogenetic trees? It's a union of polyhedral cones, and these cones are orthants. When I say orthant, I just mean you take Rn, the vector space, real numbers to the n, and you're looking at all the vectors that have non-negative coordinates. Why am I doing that? Well, if I look at this O tau, this is the set of trees with a fixed topology tau. So for instance, this is a phylogenetic tree. Yeah, and it's got some topology. It's got some branch order, right? A branches off first, and then B, and then C and D are neighbors like this. Yeah, and so now if I want to describe, if I want to set a space whose points correspond to the set of all trees with this topology, what do? Well, I say, well, this edge, the edge that goes from the root to A, that has a length. That's a non-negative real number. There's a non-negative real number on this edge, a non-negative real number for the length of this edge, a non-negative real number for the length of this one, that one, and that one. There are 6 non-negative real numbers that I need. So if you give me a tree like this, so to describe the set of trees that with this topology, that is a non-negative orthant R greater than or equal to 0 to the 6th, because you need 6 non-negative real numbers for that. Similarly, for this one, you need 6 non-negative real numbers to describe all trees with this topology. This topology is, however, different from this topology, but some things look like they ought to be the same, right? The length of the edge that goes to A is sort of corresponds to the length of the edge that goes to A over here. Yeah, so there's a sense in which these two tree topologies are related. Think about what happens if this blue edge has length 0. When this edge has length 0, it really corresponds to this sort of ternary tree with instead of a binary splitting, it's got a ternary splitting here. Yeah, and the same thing is true if I set this edge to 0, this edge length to 0. So these two trees, the two outside ones, have a common contraction in the middle one. And so what you notice then is that the orthants corresponding to them, the orthant corresponding to tau prime over here, and the orthant corresponding to tau double prime over here, they intersect along the orthant corresponding to this tree topology over here. And so the tree space itself is going to be a union of all of these orthants for all of the various topologies, glued together in ways that reflect the fact that trees can have common contractions. That's the idea. So that's what this space looks like. It is explicitly described as a polyhedral cell complex, and it has a metric on it, but it's a singular space. It is not a manifold. It is definitely singular. And I'll show you an example of that, the simplest example of that very shortly. There are various models for tree spaces. So that's the simplest one, but then there have been more sort of nuanced ones developed over the years by various people. Now you can look at products and mixtures of these kinds of things. So I want to give you an idea of what a stratified space feels like and why it comes up in data. So I want to talk about the digit 1. This is not biological, but it's a really good example for thinking about spaces of where data lie. So this is the MNIST data set, which is just a data set of handwritten numerical digits. So if I look at the digit 1 and I ask, what is the dimension of the space of handwritten ones? You think for a little bit. All right, well, I mean, what's a 1? A 1 is just a line segment, right? Yeah, so 1's just a line segment. It's maybe got a little bit of angle, but let's just say that it's basically a length and maybe an angle. So maybe it's dimension 1 or dimension 2 for the length of the thing and what angle it's at, right? But what if you're European and you write your ones like this? It's got a vertical bit and then a little sort of serif at the top. And then the serif has a length and an angle as well. So now it has dimension 4, right? Because there's dimension, one for the sort of the angle of the main part, one for the length of the main part, one for the angle of the serif, and one for the length of the serif. Now it's got dimension 4. Well, what happens if you write your ones with the serif at the bottom? Now it's got maybe dimension 5 or 6, for the length of the serif and how centered it is or something like this, yeah? So the dimension of the space of ones, what you conclude is not that, oh my gosh, there's no space of ones. What you concluded is that the dimension of the space of ones depends on where you're standing. If you're standing near one of these plain old ones, it's just a line segment, then the dimension looks like 2. If you're standing near one of these European ones, the dimension looks 3. And if you're standing near one of these even more complicated ones, maybe try 3, I say 4, or the dimension looks like 5 or something. The point is that this is a space whose dimensionality isn't even constant, and these various dimensional components intersect in various ways that are singular. That's the idea. You can go into infinite dimensions, persistence diagrams. These are topological summaries of data sets and data objects. People in this room know a lot about that, but I'm not going to say so much about that today. The point is that these are examples of singular spaces in which you would like to be able to do statistics. Okay. So the idea is you want to do statistics. You want to try to do ordinary statistics. So you have some nonlinear space M. Think of it as the space of phylogenetic trees or something. And you want to do averages. You want variance. You want principal component analysis. You want to be able to do things like laws of large numbers and confidence intervals, which in this case are actually regions because they're higher dimensional. You want a central limit theorem. And I'm going to tell you what that feels like, the central limit theorem. That's the goal for today, is focus on the central limit theorem. That's how means of large samples behave. So history for smooth manifolds, this was done relatively recently. You'd think this sounds classical, but in fact, it was done in the last 22 years, 23 years, something like that. And then there are various versions of this smooth central limit theorem. And I'm going to tell you what that smooth central limit theorem says. That's going to be, before I tell you the singular one, I'm going to tell you the smooth one. So I'll describe this in quite a bit of detail. And then for singular M, there was proved on a bunch of special kinds of spaces which are made of flat pieces, including the phylogenetic tree space. That was one of the examples. So let me show you what these spaces look like, because they give you an idea of what singular spaces look and feel like. So here is the simplest possible singular space. Why do I say simplest possible singular space? Because the space that, so if you have a one-dimensional space that is just as an arc, then that's a smooth manifold. The only way to make it non-smooth is to have some sort of branching. And so there's the simplest space that has a branch point over here, and this makes the space not a smooth manifold. Okay, so you could also take the product of that with a real line, then you get what's called a spider. Oh, sorry, you already started with the spider. This is the open book. Why is it called an open book? It looks like an open book. Literally, it looks like an open book. Yeah, so the point in the middle here in the spider becomes the spine of the book. This is an isolated hyperbolic planar singularity. We call this space the kale. Kale is a vegetable that's very negatively curved, and this is a space that is negatively curved. It's just isolated hyperbolic planar singularity. It's just too many syllables. But anyway, the point is that the space is metrically flat everywhere, except for this pink point. At this pink point, the space has sort of more than 2 pi's worth of angle glued there. So the angle sum there is more than 2 pi. So this is a singular, topologically, this is just a plane, but metrically speaking, it's singular at this point. Okay. Now here is tree space T3, right? This tree space T3 is just 3 spider. So this is the phylogenetic, this is the set of how many phylogenetic trees are there with 3 species. You should check there are 3 phylogenetic trees with 3 species, and it all depends on which one diverged from the other 2. Yeah. And so that space of phylogenetic trees on 3 species is just this T3, it is singular. Here's the space T4, and what you'll note here is that it's got a bunch of pentagons, but this edge C is supposed to be glued to this edge C over here. So this ray A is glued to this ray A back here, and this ray B is glued to this ray B. There's no way to draw this in R3. This is actually the cone over a Peterson graph, if you know that kind of combinatorics, but there you go. Okay, singular spaces that we'd like to be able to do central limit theorems on. So the goal today is to do central limit theorems on non-smooth spaces. Okay. So what does the usual central limit theorem say? The input here is a vector space, R to the D, and what you have some independent random variables, x1, x2, and so on and so forth, and they're distributed according to some measure or some probability distribution mu. And the idea is to compare the empirical mean. So what's the empirical mean? Well, x1 through x1, x2, x3, these sorts of things, these are random objects. These are random points in Rn. They're random distributed according to mu, but they're random. So if I take an average of a bunch of random points, what I get is a random mean, right? So the mean of n random points is a random mean. So this mean mu n bar, this is a random object. So this itself is another random variable. Right? This is the mean of a sample of size n. But a sample of size n is a random thing. So the mean of a sample of size n takes different values depending on which actual events you had. So the various samples of size n give you different means, and you can draw where they occur, and it kind of looks like this. Yeah, they kind of center around the population mean. So the population mean is the mean of the actual distribution. That is a fixed point in RD. Yeah, that's a fixed point in RD. And these empirical means, they sort of they sort of fluctuate around this population mean. So the law of large numbers says that in fact, as n goes to infinity, when you let the sample size get big, these sample means, they collect around the population mean. But it says that's what the law of large numbers says. So what does the central limit theorem say? Well, the central limit theorem says, well, as these empirical means are collecting around the sample mean, around the population mean, let me rescale out the set of those empirical means. Let me rescale them by a factor of root n. So the empirical means themselves are distributed according to this it's sort of getting closer and closer to a delta function at mu bar, because they're all collecting around this one point. But now I'm going to rescale them out by a factor of root n, and I'm going to get this kind of distribution. And this distribution should look to you like a bell curve, a Gaussian, and that is in fact what the central limit theorem says. It says that as n goes to infinity, this rescaled difference between the empirical mean and the population mean, it converges to a Gaussian distribution. And that Gaussian distribution is centered at the origin because you've taken here mu n bar minus mu bar, right? So you've made the origin where mu bar is, essentially. You've translated it that way. And it's got the same covariance as the original distribution itself. So this is what the usual central limit theorem says in a linear setting, one-dimensional linear setting, actually. Now let's talk about what happens when you try to go to a nonlinear setting? What fails? Well, already at the very first bit, you can't take an average, a sum or an average of points in a manifold. You just can't do that. It doesn't exist. There's no linear structure. You can't divide by n. You can't add 2 points. And there's no such, so what is a Gaussian on a manifold? Like, we know what a Gaussian on a vector space is. What's the bell curve on a manifold? You can try to get some workarounds. What do you do instead? So for the sum or the average of points, what you do is the following. You can take so here's what's called the Frechet function. So what you do is so imagine you have a finite set of points, right? And you're looking for a point in the space that minimizes something. So you've got your space M, and you've got a point in there, and so this point, and you want to see how far it is from all of these data points. What you do is you take the distances from this point to each of the data points. You square all of those. That's what this dxy squared is. And then you sum them up. That's what this integral says. And then according to this measure, so the measure would have like a 1 over n or something like that. But you sum up the square distances to all those points, and you want to minimize the sum of those square distances. That's the idea. And it turns out that in the Euclidean setting, that returns for you the average. So this least squares minimizer, this least squares approximation is the average in a linear space. But it can be phrased in this nice it doesn't require any linear structure to phrase it this way. It just requires you to be able to integrate on M. So it requires a measure. Okay, so there's still an empirical mean, mu n bar. You can still see where the empirical Frechet means lie. And they still converge to the Frechet mean of the distribution that you started with. So that's good. So the law of large numbers still remains the same. So that's what you do for number 1. You replace averages with Frechet means, which are least squares minimizers. And then for number 2, what do you do for Gaussians on M? Well, what's the defining property of a manifold? I'm going to get to pictures of this in a second, actually. But the defining property of a manifold is that it's locally approximated by a vector space. So if you know that there's going to be a population mean at a fixed point, then the central limit theorem will talk about Gaussians on the vector space that's the tangent space to that point. I want to convince you that that should be the case, but it actually is the case. So why is that the case? We're going to recast the central limit theorem on a manifold as something linear. So why is this? So when you take rescaled differences, so remember, this is what you do in the linear setting. You take mu n bar minus mu bar, and then you multiply by the square root of n, yeah? So you can't do this minus, and you can't do the multiplication. But think of what happens as n goes to infinity. These empirical means are approaching the population mean mu bar. So in the limit, what you're really saying is it's going to give you a direction, yeah? And that's going to give you a point in the tangent space, which you can, in fact, multiply by the square root of n. So what I want to do is I want to say, so this limit is going to be a random tangent vector in the tangent space, because you have points that are approaching, but then you're rescaling them out by a factor of root n. Yeah, so here's what's going on. The logarithm map, what is the logarithm map? It's a map from M, the sample space, to the tangent space. And what it does is it takes each point to a tangent vector whose direction is what you think it should be, and whose length is the distance to that point. So the idea is if my point X is down here in the manifold, then what I do is I say, well, if I'm standing at the Frechet mean here, mu bar, at the population mean, and I want to know how to get to X, what I say is go in this yellow direction for this amount of length. The length of so you go in the direction of this tangent vector. What that means is you go this distance, the length of the vector, in this direction, the direction of the vector. So the vector is instructions for how to get from u bar to X. That's what the logarithm map does. It takes X to instructions for how to get to X from u bar. Okay, so back to the linear setting. So if you have your measure mu on your space M, then it gives you a measure nu on the tangent space, and that nu is the push forward measure. Okay, what am I saying? Push forward. Don't look at the symbols. Just look at the picture. If you had a bunch of mass on this space M over here, what you're going to do is place the mass on the tangent space according to this rule, which tells you that the mass that sits at this point is in this direction and that distance along this tangent plane. So you just move the mass from M onto the tangent plane by using the logarithm map. That's what it says. The symbols can be, you can look at the symbols if you want, but that's what they're telling you to do. If your manifold has a folding, it's not a manifold. It's smooth for the time being. It's not singular yet. Are you asking, are you asking what happens if the logarithm map is not well defined? So we'll get to that. So it can have a cut locus. So what happens if you're on a sphere, then you have a point at the antipode, right? Then the antipode doesn't know which direction to go. So what you do is you make an assumption that there's not actually any positive mass at the antipode. So then so measure theoretically, the logarithm map is everywhere defined. That's what you do in that setting. It's a good question. It's really important. It's the sort of thing you gloss over in a colloquium, but it's great to get the question, actually. Yeah. Okay. So you have your measure on M, and you can get a measure, yes?
Here, your push forward of the measure, it's only local.
It's only local. No, it's why? So the push forward measure is global because the log map is, I mean, except for on the cut locus where it's not defined, the log map is defined everywhere else.
Okay. Oh, okay.
Yeah, so the question was, why? So it looks like the push forward is only local, but in fact, the log map is well defined. Exponential map is not going to be always defined, but the log map is, except for on the cut locus. Is that good enough? Yeah, okay. We're going to make a hypothesis in the singular setting. It's going to be called the localized hypothesis, which is going to take care of that anyway, actually. So now you have your push forward measure, and now you can just take the central limit theorem for this measure on this tangent plane. Right now you have a linear setting, you've got a central limit theorem there, use the central limit theorem there, and what you get is that, you know, the square root of n times this new n bar, it approaches some Gaussian distribution. So the question is that the manifold CLT? You'd like it to be, right? You've reduced everything to a linear setting in a nice way, but the answer is not quite. There's a correction term. Why is there a correction term? Very simply, it's because manifolds are not flat. So there was some curvature that you forgot. When you pushed the measure forward onto the tangent plane, you specifically got rid of the curvature. And the curvature has information that you've lost. And so you have to recover what information you had there. This is by something called a distortion map. It is the inverse of the Hessian at the population mean of the Frechet function. And I can write down the formula, but let me tell you, I look at this formula with just as much sort of opacity as you do. I look at this and I don't understand anything. This is the inverse of the Hessian, I'm sorry, the inverse of the Hessian. But what it actually means, this was sort of Tran, the at the time graduate student, was the one who really saw what this should be and explained to us what was going on. So this is what accounts for the fact that passing from M to its tangent plane forgets the curvature. And what it does is it accounts for the fact that rapidly spreading geodesics that exit the population mean can tug the covariance. Let me show you what's going on in the picture. Yeah, suppose that M looked like this, right? Suppose that geodesics through the population mean looked like this, right? So they're kind of bunched together on one side, but then when they go through, they sort of splay out really fast. Now think about what happened if you had data points sitting, a mass sitting at those, or data points sitting on those curves, right? When you take the exponential map, right? So remember, at this point, this geodesic is moving just sort of barely up a little bit. And so in the log map, it looks like it's sort of along this circle. But in fact, in M, this point is going to end up way up here. So this point way up here, when you take the log map, ends up sort of in a nice sort of, you know, they're much more bunched together in the tangent plane than they were in the original space. And this causes the covariance to squeeze toward the population mean. And so this inverse Hessian at this point is exactly taking care of this squeezing that's going on. Okay. So if you have any random variable, then you can push forward under this distortion map. And the random variable that we're going to be interested in is, of course, this Gaussian, this n0 sigma that we had from the previous slide. So the manifold CLT says exactly this, that when you take this, let me look at what this says. You take the, at the population mean, you take the log of the empirical mean. Now you can rescale by a factor of root n. And this converges to not just the Gaussian you started with, but it's pushed forward under this distortion map. So that's what this the manifold CLT says. Yeah, where n0 sigma is the limit law for that push forward measure. So compare that to the linear CLT, right? The linear CLT, these sort of drawn to be compared, right? So instead of taking mu n bar minus mu bar, what's that? That's really the vector that points from mu bar to the empirical mean. That's what this yellow minus purple is. It's that vector that goes from purple to yellow. That's what this log is. It's the vector that goes from purple to yellow. And then you multiply by root n. That's the same in both cases. And here it goes to a Gaussian. Here it goes to the push forward of the Gaussian under a distortion. Yeah.
For that manifold CLT that you're mentioning, this coordinates of n times log. The mapping that is happening in there, if I'm going to be thinking about it in terms of the data, what I need to do is mean center the data, get the Hessian of it, and then the multiplication of this should get me to the space that I'm interested in, which would be that Gaussian space?
Yes, kind of in the limit. The thing is that you don't know what mu bar, what sort of, you don't, in statistical situations, you don't know where mu bar is to begin with. And sort of, you have to sort of take large sample limits. But yes, that's the idea. That is the idea. It's just you have to do that sort of for large n and sort of all at the same time. Yeah, that's exactly right.
So is the push forward still Gaussian? Probably not Gaussian.
It is, because this is a linear map.
Oh, H is a linear map.
Yes.
So then you can, given a concrete H, you can actually compute.
Yes, this can actually be computed and is in examples. Yes, yes, that's right. Okay. All right. So in the singular setting, what happens? Okay. So now you need appropriate classes of spaces and measures. You need analogues of Gaussian random variables as limiting distributions. You need to reflect the geometry of the curvature in the limiting distribution. Yeah, so we need to take care of a lot of things. So these are the problems. What are the solutions? So this solution, number one, has a lot of words in it, and I can spend as little or as much time as you want telling you about these words, but smoothly stratified metric spaces of curvature bounded above. I actually have a slide about that, and I will say something about it. And the hypotheses on the measure are that it should be localized. That's Francois's question. It should be immured, which is like, I'll talk about that. It has to do with where the empirical means sit with respect to the support of the measure, more or less. They should be in the convex hull, kind of. Amenable means that you should be able to use Taylor series, so it should have enough derivatives to be able to do that. And it should be a measure on M. Okay, that's mathematics is so what's the point? Mathematics is all about formalizing what we think ought to be true by writing down the hypotheses that need to be true in order to be able to prove that it is true. So the fact that there are precise hypotheses that come up here should be comforting and not a bad thing, right? It's comforting because it tells us what we actually needed in order to prove the central limit theorem. That's what these hypotheses really are. They are the indications of what came up in the proof. And frankly, what can fail when you don't have these hypotheses, maybe the central limit theorem isn't true. So you shouldn't try to apply it in some situation where you don't have any handle on some of these things. That's the idea. So then we're going to reduce to the linear case, but because we're in a singular setting, that takes an extra step. Here's the idea. There's going to be some tangential collapse that goes from the tangent cone to r to the m. Tangent cone, whoa, wait a minute. So remember, your space now is not smooth. The defining property of a smooth space is that it is locally approximated by a vector space. So if you have a singular space, then quite specifically, it is locally approximated by, well, whatever it's locally approximated by, it's not a vector space. It turns out to be a cone, because if you leave a point, you can always leave 10 times faster or 3 times faster or half as fast. you can multiply tangent vectors by positive real scalars, but the set of vectors is not a vector space. It is a cone instead of a vector space. It's some singular thing with a singular point at the origin. So we're going to call it a tangent cone from now on. It is not a convex cone in that sense. It's just closed under positive scaling. So the idea is you take M and you replace it with its tangent space. That's something we did in the smooth setting. But now in the singular setting, the tangent cone is singular, and now we have to further collapse this tangent cone onto a vector space. Then we're going to put a Gaussian on this thing, right? We're going to put a Gaussian on this vector space. It's actually going to be supported on some subspace. And then we're going to somehow lift that back to the manifold again. So this is the same plan as the manifold setting, except that in order to get to a linear space, there's 2 steps instead of 1. In the manifold setting, you take a tangent space, we're linear. In the singular setting, you take the tangent space, that's now a cat 0 cone, and then you have to flatten that somehow onto a vector space. Then there's the distortion map, and that's going to be actually the same kind of thing. But this distortion map is going to be really interesting because, do you remember the distortion was defined as the inverse of a Hessian? The problem is that that Hessian becomes singular. So it doesn't have an inverse. But the inverse Hessian limits to something nice. So you should be thinking of the inverse Hessian the whole time, and don't think of it as the inverse of the Hessian. Think of it as this thing that's the inverse Hessian. And the inverse Hessian has a nice limit. Again, that's Doe's insight there. So the singular CLT is the same statement. This is, I cut and pasted the tech, right? This is verbatim the same statement that I wrote in the smooth setting. It's just that the meaning of this H lower sharp is different. Everything else essentially means the same thing. Yeah. So it's the same expression, and this tangential collapse is hiding in both the limiting measure and in this Gaussian and in this distortion. I could tell you a little bit of hypotheses. Okay, so M should be a nice union of finitely many manifolds. So I want to tell you what kinds of singular spaces we're able to talk about, and that's going to be this slide over here, stratified spaces. So this is just to give you an idea, sort of, let it wash over you what kinds of spaces we're thinking about. I want M to be, okay, it's going to be complete, locally compact. It's going to be a geodesic space. That means that the distances between points are realized by actual paths. And I want M to be a disjoint union of strata. Okay, so each stratum is going to be a manifold. It's going to have its own distance on it. And the closure of each stratum is going to be a union of previous strata. So the examples will tell you what I'm going on here. So what's going on? So the example of a graph or a network, here the strata, they're supposed to be member manifolds. So you take all the vertices, those are strata of dimension 0. And all the edges, those are strata of dimension 1. And the actual example is a fruit fly wing. Yeah, so here's a fruit fly wing. This is a graph. And so the strata here are the vertices. There's one. There, oh, sorry, people online, right? Here's a vertex. Here's a vertex. Here's a vertex. Here's a vertex, vertex. So there's like maybe 12 vertices or something like that. And there's maybe, 20 arcs or something like that. Yeah? Okay. So that's what or you could consider this actually if you wanted as a 2-dimensional stratified space where you have vertices, arcs, and the regions in between, which would be 2-dimensional strata if you wanted. You could do that too. Here's a polyhedron. The strata here are relatively open faces. And the example is the tree space, where here now the strata are, well, there's one stratum for this point back here. There's a stratum for each ray. There's a stratum for each 2-dimensional face. Yeah, so this is going to be a 2-dimensional stratified space. And there's these shape spaces where the strata are abstract to the equisingular loci. I can tell you about that if you want to know. Okay, and you want it to have locally defined exponential maps that are local homeomorphisms. What that means is if you're standing in the tangent cone and you give me a little ball in the tangent cone, then I'm able to say that little ball in the tangent cone corresponds to this open subset of my original stratified space. That's the idea. So that's the local exponential map. If you give me vectors that are too long, like even on the sphere, if you give me a vector that's too long, you can exponentiate it, but you'll sort of start wrapping around. Yeah, so in singular spaces, it's possible for you to simply not be able to exponentiate a vector sufficiently far, because there might be a boundary, right? And you can't leave the space or something like that. The curvature is supposed to be bounded above by kappa. that's a condition that's precise, but it essentially means that triangles are sort of not infinitely fat, whatever that might mean. I don't want to describe it, but if you want to hear about curvature kappa, I can tell you about that later. But tree space is cat 0. Usual flat spaces are cat 0. The sphere is cat 1. So the triangles are fat, but not arbitrarily fat. They're sort of like a sphere of radius 1 is cat 1. The sphere of radius r is like cat 1 over r, I think, something like that. Okay. So there should be stratified spaces. These locally well-defined exponential maps, that allows you to bring the asymptotics of sampling sort of to and from. They allow you to change between M and the tangent space at the population. That's what the local exponential maps allow you to do. And the curvature bounded above by kappa, this is not really a condition. If the curvature was infinitely positive, then the Frechet mean would run away from it so fast that you'd never actually see it in nature. So, and it's also only needed at the population mean, but it's also not really much of a condition. It's needed for the math, but statistically speaking, you'd never see a Frechet mean with infinite positive curvature anyway. Hypotheses on mu, I've already sort of summarized what these things are, so I won't go into detail. Again, amenable is a differentiability condition. Immured is a sort of geometric, empirical means lie in convex hull of the support kind of condition, which might be always satisfied. It actually, we don't know. We have a conjecture that in fact every measure is immured. We just can't prove it. We don't have no counterexamples, certainly. Localized, this is saying that you can take log maps. So the log map is almost surely defined, and the Frechet mean is unique and that sort of thing. Singular distortion, what can the distortion actually look like? Yes, Francois.
What do you mean almost surely defined? You mean with respect to the Lebesgue?
What does almost surely defined? It means with respect to the given measure mu. So I just repeated the question, is what does almost surely mean? Almost surely means, well, with respect to the given measure mu. So you've got a measure on M, which what we mean is that the mass on the cut locus should be 0.
Yeah, but it was with respect to which measure?
Mu, the one that's given. So we have our space M, and it has a measure on it, and we want the cut locus to be measure 0 with respect to that measure given. Okay. What can the limiting distribution look like? So I want to say what the outputs can look like in singular settings. So the example I want to give you is this scale, this isolated hyperbolic planar singularity. So this is described mathematically as the cone over a circle. So this is the surface. It's a two-dimensional thing, but this circle has length bigger than 2 pi. Yeah, that's okay, fine. It's a circle of length bigger than 2 pi, but I want to take a point now that's at distance 1 to all points on this circle of length bigger than 2 pi. This can't be embedded in the plane. Well, it can't be embedded in the plane. It can be embedded in 3 space, and here's what it looks like. It's the same picture I drew before. It's just the same picture, yeah? So the point is that the angle sum at this point is bigger than 2 pi, because if you go from here and you go out at distance 1 and then you draw a circle, the circle has, in this case, length 7, what is it? 7, 1, 2, 3, 4, 5, 7 pi over 3. Yeah? So it's bigger than 2 pi because there's 7 equilateral triangles glued together at that point. Yeah. And note, this singularity is geometric, not topological. This is still homeomorphic to the plane. The push forward under the distortion map is a convex projection. And here's what it looks like. It looks like this. So this is what distortion does. Distortion pushes everything onto this cone here. This capital K is called the fluctuating cone. What do I mean by that? Well, let's just say what I mean. So think of this picture where you have all these diverging geodesics. Imagine what would happen if you took the limit as the curvature went to sort of negative infinity here. Negative infinity is okay. And what happens is that now this one geodesic splits into those. Think about it the other way. If I have a point that's sitting over here and I ask what's the shortest path, the shortest paths for all of these points pass through this one. And what that means for this distortion is that it gets thinner and thinner and thinner and thinner until it collapses, boom, onto this cone. And that happens for this distribution, where what you've done is you've taken 5 points and you've sort of separated them sort of evenly. But because of this space, having all this extra angle over here, there's this big shadow over here, and that shadow is causing this geodesic collapse, this convex projection onto this cone. That's just some idea of what this sort of thing looks like. So the limiting distribution, what it's going to be is you take a Gaussian on the plane, And then what you do is you restrict it to this cone. So you're going to have a Gaussian piece sitting right over here. Oops, zoom. You're going to have a Gaussian sitting, a piece of a Gaussian sitting right on this cone over here. And then on this ray, what you're getting is the projection of a Gaussian. So you're taking the same Gaussian, you're just projecting all that mass onto this ray. And for this point, you're taking all the mass in the Gaussian that sat in this cone back here, where this is a right angle with that edge, and this is a right angle with that ray. Yeah, and you take all that mass and stick it onto the origin. So the limiting distribution has an atom at the origin, a singular distribution along this ray, a singular distribution along this ray, and a nice a non-singular distribution along the interior of that cone. That's what the limiting distribution looks like. Okay. I promised in the abstract I'd say something about singular Gaussians. This slide is very imposing. It's imposing to mathematicians and biologists alike. It has sort of, if you take our main theorem and you just sort of step back and say, well, how would an analyst rephrase that in terms of, you know, spaces of measures and sorts of things, then this is how these things would get rephrased. I'm not going to spend a lot of time talking about this, but I do want to talk about the second one a little bit and just sort of tell you, to give you a flavor for what these kinds of statements say. Okay, so what is the central limit theorem? The central limit theorem is going to be this statement up here. So what's on the left-hand side is the same expression that you saw on the left-hand side before. What's on the right-hand side is a directional derivative. So what it is, it's a directional derivative at mu. Okay, so we have the space of measures. Think of the space of L2 measures on M. Yeah, so mu is one of those. It is a measure on M in its integrable in the appropriate sense, right? So now standing at that measure, I want to ask, how can I try to deform that measure? Okay, so I'm going to try to, well, okay, so what function do I want? If I take this space of measures, P2 of M, so these are all L2 measures, each of them has a barycenter, yeah? So this is, what it does is it takes each measure and it replaces it with its Frechet mean, yeah? So that's a function. It takes measures to points in M. So it's a function from the space of measures on M, that's this, to M itself. And it just takes each measure to its Frechet mean. Now I can ask, when I'm standing at the measure mu, I can ask how the barycenter changes when I try to deform mu by adding a random point. So for those of you who've seen, what are they called again? Robust statistics. I was just talking about this. What were they called again?
Influence functions.
Influence functions. These are influence functions. Yes. If I have my measure and I take the mean of it, but then I add a point over here and I ask what happens to the mean. That's the influence of this point on the mean. Now, what would happen if this point weren't just a point, but were a random point? Then what I get is a random influence. What happens if this point is not just random, but Gaussian distributed? Then what I get is the influence of a Gaussian point, adding a Gaussian point to my given measure. Now, in the linear setting, this is dumb, because if you add a Gaussian point to a measure on a linear space, it deforms the measure in a Gaussian way, which is an obvious statement. But in a singular setting, the deformation is really interesting. And when I go back to this picture, essentially what I'm saying is if I try to, so the Frechet mean is going to sit at this point. The Frechet mean of this measure of these 5 points sits at the origin over here. If I try to perturb this measure by adding a point that sits up here, it will move until it hits this boundary and then it will stay forever. It will not leave this cone. Similarly, if I perturb by adding a point over here, it will stay at the origin and will not proceed. But if I perturb by adding a point over here, it'll move in that direction. So this this perturbation, this influence function view of, so you're looking at Gaussian, as an influence function applied to a Gaussian random point. And so you want to see the influence of a Gaussian random point on your Frechet mean. That's what the central limit theorem is saying. Okay, biologically, what's happening? Recall, remember distributions on tree space, they come from all sorts of places. In particular, they come from reconstruction algorithms, or they come from medical imaging, when you're looking at blood vessels, lungs, nerve cells, all sorts of branching structures and so on. So there's an observation which I want to highlight here, just that's about singular central limit theorems. If you take this very simple tree space, which is just a tripod or the spider, whatever you want to call it, and if you put 3 point masses just on the rays, 3 point masses like this, yeah? So what is the average of these 3 point masses? Everyone sitting here is going to tell me that the average is the origin, and that's correct. By symmetry, right? It has no choice. It can't be on any one of the rays because which one would it be on? It's symmetrical with respect to that, yes? Now what happens if I wiggle these points? Let's say I take this point over here on the, let's take the one on the top and move it up by epsilon. Where does the Frechet mean go? Well, you'd like to say that it moves up a little bit. In fact, it does not. It stays put because there's 2 points weighting it down and only 1 point weighting it up. So you have to move this point up here twice as far out before the Frechet mean starts to move off of this stratum. Yeah, so that's the thing. To pull the Frechet mean off of a lower stratum in tree space requires a sizable sample bias, an actual sample bias before you pull the Frechet mean off. So what that means is if you try to do statistical inference with all these trees, all these phylogenetic trees, the Frechet mean in tree space, if you find a tree that isn't just the star tree, which says that, yes, those are all species and they have a common ancestor, that tree would just be, there's the common ancestor, and then all of them diverge from that. That's not useful information. If you see a tree that has actual information about some, divergence, some of these species diverged before those, if you see that in the population, in the empirical mean, then that was really strongly present in the data. That's what you get from this. Because of the stickiness in this singular setting, when you see those patterns in actual data, they are very strongly present. Now, by the same token, if you try to do this process on these sort of geometric trees, where you sort of take your geometric tree and you pick a landmark, you have all these, the landmarks are given to you, and you join each landmark to sort of like the closest point in the tree, that manufactures a phylogenetic tree out of a geometric tree. That phylogenetic tree is not useful information because essentially the process that generated it was geometrically random, and you don't care whether this lung vessel branched off before that one. You care that one branched that way and one branched that way, but you don't care in which order they did that. And so you're getting kind of useless geometric information. So here you get the Frechet mean is off in the star tree. Okay. That's all I want to say about biological conclusions. Going forward, there's lots of things to do. There's interpretations of Gaussian objects on singular spaces in terms of heat dissipation, random walks, infinite divisibility of probability distributions. Statistically speaking, so now we have a central limit theorem, but now you want to do statistics, you need convergence rates, confidence regions, you want to be able to do some smoothness or singularity testing. You can actually say, let's see, I have my data, I want to know whether it's sampled from a smooth space or a singular space. Try to use the central limit theorem where you know what the convergence looks like, and if it looks like it's not linear, well then it's not a smooth space. That's something you can actually make a test to determine. You can try to learn stratified spaces, and I've already talked about singular influence functions. There's infinite dimensional singular settings. Again, I'm going to leave those for the mathematicians who know about what these things are. Curvature invariance. From distortion and tangential collapse, the types of pictures that I was drawing, they sort of generalize the two-dimensional angle deficit, right? Because what was coming up when you had that sort of that convex projection was some reflection of the angle deficit. How much bigger than 2 pi was that circle is the question. Yeah, and so this gives you sort of a probabilistic way to measure some generalization of angle deficit. You can try to integrate these things and compare with singular homology or intersection cohomology. These give you essentially, the idea is if you have a singular space, probe that singular space by sampling from nearby the singularity and see how the Frechet mean behaves nearby the singularity, and then try to recover, say, topological information about the singularity given by reading off what happens in the limiting distribution for sampling from nearby that singularity. That's the idea. This is all looking forward. I have no papers about this, but I would love to. This last one, so distortion somehow corresponds to how a central limit theorem transforms under a morphism. And the proposal is for a real complex variety. Take a resolution of singularities, take a central limit theorem on now that smooth thing, push it forward to the singular thing, and then there should be some correction term like distortion. Distortion is a correction term, but there should be some correction term that accounts for singular spaces too. And it should involve some local sheaf theoretic data around the Frechet mean. And then the conjecture is that this results on a well-defined central limit theorem on X. And there's an analog. People did the same thing for multiplier ideals. Multiplier ideals started off as an analytic thing where you sort of did some integration nearby some singularity. And it turns out that you can get the same thing by taking a resolution of singularities, taking some sheaf theoretic data and pushing it forward. I'd like to do the same thing for central limit theorems so that we can get central limit theorems on much more general singular spaces than we're able to deal with right now. And this would give you some really interesting applications to asymptotics of sampling from moduli spaces, which are, after all, basically singular algebraic varieties. And it'd be really nice to know what sort of average behavior of random deformations look like. That's what the question is. What's the average behavior of a random deformation? I would like to know that, but I have to know I have to know this stuff in order to know this stuff. Yeah, that's the idea. And with that, here's some references, and thank you for your attention.
Thank you very much. Before we start the questions and comments, I first apologize for a little bit of noise and disturbance. In the beginning, there was a little bit of miscommunication with one online participant. And thank you for running through the talk without much of, without making it possible. The second thing is, like, the ceiling mic is on. So if you want to ask a question, kindly go ahead. And for the online participants, if you want to ask a question or leave a comment, either put it in the chat box, I read it out, or unmute yourself and do so. Yeah. Time for questions. Please.
You mentioned that there were some singular spaces you wanted to deal with by resolving the singularities. In that last slide.
Yes.
What kind of
Algebraic varieties. So these what do we call them? Smoothly stratified spaces. When you get down to what's allowed and what's not allowed by these things, let me tell you something that is allowed. Crossing lines is allowed. Let me tell you something that's not allowed. If you take a plane, and from it so inside that plane you have a parabola, and take the parabola and everything above it out. So you just have a piece of a plane.
Yeah.
That is definitely a stratified space. It's got the one stratum and then the smooth stratum. It's not allowed, because the problem is when you stand, the local exponential maps don't exist. When you stand at the bottom, stand at any point on the parabola, you can look at the tangent there, but you can never reach any point sort of above this line. You can never reach it by exponentiating, because there's no tangent vectors that point up at this point. So the local exponential maps they restrict the spaces a lot. In my mind, they're more or less, they're spaces that have they're sort of normal crossings. They're transverse intersections of things. I would like to be able to do arbitrary algebraic varieties. Would be much better.
Maybe you could do subanalytic, because you have Hironaka desingularization.
Yes.
And they are roughly intersection of smooth manifolds.
Yes. Yeah. So that's right. I mean, that's what I say. The subanalytic. If I could get all subanalytic, that would make me extremely happy. I don't know what smooth spaces to use for a given subanalytic space. Are there theorems about comparing subanalytic spaces to smooth ones?
There is a version of desingularization, Hironaka desingularization. So you have a kind of blow up.
And you always get a smooth space this way?
Yeah.
Oh wow, I'd love to see this theorem. That's great news. I didn't know that, and was hoping that something like that would be true, but I've never seen it. So basically, there are desingularizations of arbitrary subanalytic manifolds.
Yeah, this is a version. This is due to Hironaka, but it's even harder than the complex.
Yeah, even harder than they were. So Hironaka's theorem has been reproved a number of times. There is now a proof that essentially is by writing down the algorithm, and that proof is only 30 pages, and it's pretty understandable. Hironaka's original proof is like 250 pages, and there aren't that many people who understand that entire proof. But yeah, so I didn't realize he at the same time did subanalytic sets. That's awesome. That's great news. Thank you.
That's an interesting talk, thank you. And interesting because the first motivation was just comparing different objects with different structures and doing statistics on them. And
Yes.
What people in machine learning do, they basically devise some embedding function, which is learned by, let's say, some graph neural network or whatever.
Yes.
Yes, and then basically move to the embedding space, which is Rn, and do all the statistics over there. And how does those approaches compare? Because is my understanding correct that some of those approaches can be interpreted as a kind of well mathematically defined embeddings?
So yes and no. So there's evidence that those spaces that you get from machine learning, more of them are singular than people had hoped. Or sorry, some of them turn out to be singular. When you can do your algorithms, and you can try to run them. If your space is singular, your algorithms might work, but they also might not. the algorithms sort of all assume that the space is smooth without actually checking anything. Yeah, so often the spaces that come up in machine learning algorithms are singular in unexpected ways. And this shouldn't be too shocking. I mean, again, with the things like the spaces like the set of ones, configuration spaces of various sorts are going to be sort of not even have a fixed dimension. And when you're looking at complicated, things like images and all that, then the sort of the tangent space, sort of the space of nearby images might be much smaller dimension when you have one kind of image than when you have another kind of image. I don't know if that addresses what you said, but I had a conversation with a student who was in the computer science department about some of the singular stuff, and he was actually about to start investigating exactly this problem of the spaces that come up in machine learning algorithms, and pointed out to me that there are papers that say that these spaces need not be smooth. I don't know what happens to the algorithms there, so I don't know what effect the non-smoothness has on those kinds of algorithms, because they're, well, let's just say I don't understand. There's a lot of nonlinearities in the algorithms themselves, and I don't know how those interact with the singularities of the spaces. So that's a big problem at the moment. Big in the sense of it involves knowing a lot of things beforehand and sort of checking what's true and what's needed. And I don't sort of have knowledge in any single one of those things.
Within this aspect that you're mentioning, somehow, because one of the things that is to some extent being introduced when it gets to this point is the introduction of the redundancy between the different, because when you're talking about all these manifolds that we're talking about, there are basically different accesses associated with each of these elements of the manifold. You have a point in the n dimension, right?
Yeah.
And then if you have several samples, that's as an example, like the ones that you mentioned.
Yes.
Yeah, and then so every one of them would be represented by this vector that has so many different points in it, and then that becomes one single, in that manifold becomes one represented. And then if you consider the redundancy that could be included, then that might, as you mentioned, the space is not smooth, it's not well defined, but that can actually give a kind of the bypass to it. And that's probably how some of these guys are.
Yes, I would view that as essentially a way of desingularizing the space. So that's kind of what desingularizations do in some sense. What they do is they remember additional data. So what the term blowing up came up before. What that really does is it says, well, when you're standing at a point, you can look at the direction that you can leave that point. And what you essentially do is you remember not just where you are, but also where you are and what directions you can leave. That's what blowing up does. It replaces the space with a space that sort of remembers not just location, but location and tangent data. And the theorems about algebraic varieties are that when you do this cleverly in the right way, you end up with a smooth space after finitely many steps. But in any given application, remembering enough redundant information might give you enough of a desingularization to be able to make your algorithm work. Agreed. Yes. It would be the issue of detecting what singularities you have and what kinds of redundant information you need.
Actually, this is the thing that Naftali Deshmukh, he passed away a few years ago from, I think, about Tel Aviv University. I don't really remember it. But this was the thing that he was working on with the information theory approach. And some of the things that he showed that why some of these deep stuff are working and why they're not.
That sounds awesome. I would love to get a reference from you after. Maybe it was Ben Gurion University or something like that.
It was actually the last NeuroIPS, which, I mean, not the last one, but the last of his period before he unfortunately just.
Okay, yeah, I would love to get a reference.
2019 or 2019 most probably, before the COVID. And he had amazing, beautiful. He collaborated with this dude, I forgot his name, at MIT, contraction theory. He has. I
don't know this.
I'm terrible with names.
We'll figure it out. We'll figure it out. We'll figure it out. Yeah.
He has a French kind of taste in that last name.
Okay. Cool. Yeah. I'll come up to you and we'll figure it out. Yeah.
Is it Rigolet from MIT?
Could be. Honestly, I've names never.
The guy doing high dimensional statistics.
Probably. He actually visited us a couple of times. This
okay, that'll make it easier to find out who it is.
made some connections between different visitors.
Yes, exactly.
Very good. Thank you for the comments. Further questions or comments?
Anything online?
Including the online friends. If that is not the case, before we thank Ezra again, I wish to point out that he'll be around until the end of next week. Next week will be the symposium of our program, hence a little bit busier. If you want to talk to Ezra, probably the next couple of days will be a better time, and we intentionally included the coffee time on Friday afternoon to actually stimulate discussions between visitors of our program and researchers at it. So you're all welcome to join us for the coffee time and further discussion. If there are no more questions or comments, let's finish with thanking Ezra again.